Welcome everyone to PathAI's machine learning webinar series. My name is Rob Brockett. I'm the Director of Biopharma Partnerships here at PathAI, and I'm excited to kick this off. We are today going to provide an introduction to machine learning for digital pathology, but the aspects of the things that will be covered today will be outlined in more detail in the upcoming webinars that you see here listed. If you have any other topics of interest that you'd like us to potentially cover in webinars in the future, please submit them via survey that will be following this webinar. The intent of this series is to empower our partners across pharma pathology and research and really provide an understanding of AI and digital pathology and its applications for both innovation and development. Just a couple of housekeeping things. If you have a question, please submit it via the Q&A button at the bottom of your screen, and I'll be taking notes of what questions are there, and we'll have a Q&A session at the end with Amaro. So with that, I would like to introduce Amaro Taylor-Weiner. He is our VP of Machine Learning at PathAI, and I'll kick it over to him to start the presentation.
Thanks a lot, Rob. So as Rob mentioned, I'm Amaro. I'm the VP of the machine learning team, and I'll be giving an introduction and sort of a survey to a few different machine learning topics for digital pathology. That's going to go in sort of three sections. The first section, I'll talk about the role of AI and opportunities for AI in digital pathology, at least from how PathAI sees it. We'll talk about elements of algorithm development. I'll break that into two sections. The first is going to be covering challenges for ML development in digital pathology. And then I'll cover briefly how we approach those challenges at a high level in terms of data, models, and evaluation. And each of those sections will contain topics that will be covered in more depth in next webinars. And sort of set some of those up. And then finally, I'll end with considerations for choosing models and evaluating approaches for external vendors and just ML and digital pathology in general. And as Rob mentioned, please drop questions in the Q&A. I can't see you, so if you're like waving your hands or anything like that, we're not going to be able to catch that, so just drop them there. Cool. So starting with AI and digital pathology, the roles and opportunities. So we see at PathAI, we see that there are shortcomings in in pathology that are addressable via machine learning. And these are really the opportunities that we're attacking in this space. So if you think about classical pathology, the search space of hypotheses and biomarkers that you can find is driven by reliance on experts, tradition, and sort of a history of medical practice. Of course, that's great, but there's many hypotheses that a human possibly can't explore because they can't compute things exhaustively on a whole slide image involving millions of different points. There's historically a lot of inter and intra observer variability, whereas in contrast to that, machine learning methods can be objective and reproducible, objective meaning to some standard that's reproducible. They can learn systematically, so you can set up data sets and have a machine learning model improve over time. That's difficult. The field of pathology will move more slowly than a particular model. Maybe that's good, maybe that's bad, but there's an opportunity there. And then finally, I think one of the major key points is that digital or pathology by humans is not quantitative and it's really hard to scale. Whereas for AI-powered pathology, it is inherently quantitative and it's also inherently scalable. We can sort of run these methods on the cloud. So we think that there's a lot of opportunity here and we're really going after these in particular. So how are we approaching this problem, or how do we see this problem evolving over time? So we think that because digital pathology can be involved in all stages of development, from exploratory research to clinical development through to clinical care, that there is an opportunity to really have a virtuous cycle in digital pathologies. So you could start by generating more data, characterizing your patients more richly. That makes available new hypotheses to explore. You can then find some hypotheses with high signal validate those in clinical development, deploy them broadly, and then collect data and information from new patients about where those new biomarkers worked, where they didn't work, and use that to generate the next generation of models and really fueling exploratory research. So that's where we want to go. That's the opportunity of that cycle. So how does that meet machine learning and what we're doing really today? And I think the way that we're realizing that in the machine learning team is by building our technology stack that works using like high scale machine learning operations and orchestration, high scale model evaluation, and a diversity of model architectures to build a set of products. We're deploying those products today as RUOs in our AI site viewer. That enables us to collect real world data and the interaction of sort of clinical slides through our products. We can see how pathologists respond to our products, whether they think the product is working well, whether it's not working well. That in turn generates more data, which we can annotate and source through our data partnerships and through the pathologists that we work with, which then feeds right back into our product development and enables us to release the next version of the product, and that cycle just continues. So we're really set up, and we were focused when we were developing our ML capabilities to really be able to scale and develop quickly and iterate on data and feedback that we get from the real world. Okay, so those are the opportunities and sort of how we're approaching the field. Let's now dive into some of the elements of algorithm development, starting with the challenges. Okay, so the challenges in machine learning for digital pathology, I think really there's three categories of challenges. The first category is around interpretability and explainability. So interpretability is your ability to relate the machine learning model's outputs, or really its outputs and its performance to biological objects, and so that it can be integrated with scientific knowledge or related to molecular data, for example. Explainability is the ability for your model to tell a pathologist or a clinician why the model is predicting what it's predicting, what is important in the slide image. And that's really required for physician trust and patient safety. The second challenge is around overcoming technical covariates. So as we all know in digital pathology, an image generation process, and that process can be highly variable depending on where the data was collected. So you can get variation due to sampling, maybe due to scanning, due to staining, due to tissue preparation. And all of these things are aspects that a human pathologist is robust to, right? A human pathologist can operate on many different scanners, no problem. Machine learning models operate a bit differently and can perform differently if there's noise in those images generated by these technical covariates that you're not accounting for during model development. Finally, the last challenge, and I think this one is particularly important in this space, is that there's a lot of data scarcity, and curating data sets can be quite difficult. So, in any clinical setting, it's going to be hard to get Facebook level data sets. You're not going to have billions of data points to train these models. So how do you construct a data set and construct a data set channels in order to build machine learning models that will work in this space? I think there's model architecture and modeling choices you can make to increase your power. We'll talk about those, and also some of the data partnerships that we've generated here. Okay, so now through the next part of the talk, now that we've covered the challenges, I'll talk about how we address those challenges using data, model, and then evaluation. So starting with data, in digital pathology, the core data component is really whole slide images. So what are some characteristics of whole slide images that you need to consider for model development? Well, I think that there's really two types of data features to consider. The first is variables related to the image generation. So these are things like collection type. So is it a biopsy? Is it a resection? Is this a TMA core? Is there a negative control on the slide? It's related to scanner and lab specific factors. So how was the data collected? What scanner is it? And then finally staining protocol. So where was it stained? If it's a H&E, or is everyone using the right protocol? Did histotech A do the staining versus histotech B? Considering that kind of variability in understanding your data is important. And then the second factor is making sure that you're assembling data sets that have the right patient characteristics for data splitting. So depending on the indication, this can change a lot, but things like tumor stage, grade, histological subtype, maybe patient BMI, tissue source site. So is it a metastatic sample? Is it a primary sample? These are all really important factors which will change the way your model performs. And you want to make sure that you're having those represented in your train set and your test set so that you can understand your model performance. So on the right here, I have a diagram to kind of highlight this. And you can see how the train and test domain, or those variables that I'm talking about over on the left, can influence sort of the visual aspect of the prostate cancer and also the visual aspect of the image. So for example, the histological subtype might change what prostate cancers you're looking at, or that patient population is generating the types of cancer you're seeing, as well as the scanner and lab factors are generating and changing the way that image looks. And both of those factors change the way your model is going to perform that prediction, which in this case is segmentation of tissue areas. The second part of data, after you've sort of understood and handled your input data, is what is your target of interest? What are the labels and tasks that you want your models to perform? So you want to think about what is the use case and what is the data I need to be able to learn that. So there's three different types of tasks that I'm going to cover here. The first is supervised learning. So this is pixel labeling or pixel classification. And it's typically used for quantification of tissues. So let's say you want to quantify and profile the tumor microenvironment in terms of cells. What you would do is you collect pixel annotations on whole slide images. These are typically done by pathologists who hand label cells and tissue areas. One of the challenges here is that it's tricky to generate that at scale. That's one of the things that at PathAI we worked really hard to address by assembling our pathologist network. And the other way to address it, which is more of an emerging area, is something we do here and is done in the field as well, is leveraging IHC or other imaging modalities to basically overlay on an H&E image to generate annotations at scale. So you're going to hear another webinar in this series about an approach that we've developed here that doesn't use IHC. It's actually just imaging technology called quantitative multimodal anisotropic imaging. And basically what that's enabling us to do is identify all the fibers in an image in a way that we can generate sort of super annotations at scale, similar to that you might do with an IHC. Some of the challenges there is that you have to be able to align that IHC image or the QMI image with the H&E in order to generate those annotations. So if you have serial sections, that can be challenging depending on how those cells move. There's also, you can also recover slip slides, so you can restain and then take the cover slip off and then stain again. That also has challenges due to distortion of cells. There's off-the-shelf tools that work pretty well for that alignment. We have that capability here, but it still remains a challenge. The second type of learning and labels that you might want to work on are what we call weakly supervised learning. And typically that's to classify something about the whole slide image or the patient. So some whole slide scores that you might want to predict are things like disease stage or grade, for example, like NASH scoring. Another area that you might be interested in developing is in genomic variables. So that's a characteristic of the patient's entire tumor, like trying to predict if this tumor is BRAF V600E positive, for example. And then the last type of learning that we do in this space is using unlabeled data for unsupervised learning. So all that requires is essentially a huge set of images. And the idea there is to learn representations of digital pathology images that might be useful for downstream tasks, like pre-training a model to work for multiple instance learning, or developing unsupervised representations for downstream biostatistic or bioinformatics analysis, or connection to molecular data sets. And at PathAI, we've worked hard to develop data streams that kind of address the previous slide, but also this slide in terms of our ability to develop these models. So we have our own diagnostics lab in Memphis where we can source tissue blocks and slides. We also have academic medical partners where we get slides, molecular data, clinical data. We have reference lab partners where we can collect molecular data with images. And then we also have a data marketplace where we can get some real world data with clinical outcomes as well as molecular data to tie into the rest of those data sets. Okay, so that was data. Now quickly moving into models. There are many types of machine learning models used in digital pathology. I've kind of summarized our zoo of models, our model zoo at PathAI, in 4 different categories. And I'll talk about each one briefly. So we have CNN, or deep convolutional neural network segmentation models. These are typically used for supervised learning and pixel classification. So these are the workhorse models that we use for our human interpretable feature models. We have graph neural networks and our end-to-end models. The end-to-end models are also deep convolutional neural networks. They're just not just for segmentation. These, the GraphNet models and the end-to-end models both do whole slide classification. So that's that weakly supervised learning that I mentioned in the previous slide. We apply the GraphNet models to develop spatial signatures or learn spatial models on top of our segmentation and cell heat maps. We also use those for multiplex immunofluorescence. You'll hear a lot more about that in an upcoming webinar as well. Same with the end-to-end models. We've developed our own in-house formulation of multiple instance learning that we call additive multiple instance learning that we published at NeurIPS last year. We think that that's superior to the traditional attention multiple instance learning models. You can definitely hear more about that in the webinar or check out the paper. And then finally, the last type of model that we leverage is generative models. So these are unsupervised models where essentially what we're doing is using them to generate new digital pathology images as a way of supplementing our data sets. So we have 2 generative models that we use in our product development all the time. Those are scanner and stain transform models. You can see the scanner transform model on the top. In that top row, we have a single slide that we've scanned on 4 different scanners. And on the bottom row, we have the synthetic image that's generated to try to mimic the effects of scanning on different scanners. So we use that to really expand our data sets and generate more robust models. And then we do the same with staining, where you can simulate sort of different staining protocols or the variability that you might see in staining in the real world. Okay, so how do we combine those models to make them into digital pathology products? So most of our, or all of our digital pathology products, all the products that we build at PathAI, are essentially a combination of models from that set of architectures. And we bring them together in a modular fashion to create a product that solves a particular problem in the space. So I have some examples of how we might plug together these blocks to create a new product on the right, and I'll talk through them briefly. But yeah, so here we have an example which might be like a genomic triaging product. So this is something that's going to predict a molecular marker from a whole slide image. So that model or that product is created from 3 different models. The first is an artifact classification model, which sort of maps out the usable tissue in the whole slide image. We combine that with an additive MIL model, which we run after the artifact model to do the whole slide classification. And we train both of these models in the setting of scanner transform. And what we get at the end by combining these 3 units is a scanner robust method for predicting some genomic marker. We also can do this for NASH. So let's say for NASH, you wanted to make a NASH staging product that works solely from H&E. You could do something where we do synthetic staining. We start with generating a trichrome image from H&E. Then we apply artifact classification, then tissue segmentation, where we label every pixel what tissue that is. And then we apply a graph neural network on top of that to classify each biopsy as a NASH grade. So it's doing that NASH staging. We can also do that for quantifying the tumor microenvironment. So here I have an example all the way on the right for quantifying the tumor microenvironment in non-small cell lung cancer, where we combine an artifact model, nuclei segmentation, cell classification, and tissue classification to create a set of human interpretable features that are going to be useful for translational research downstream. So this is really the model that we use to create our pathology products. We've built a system and a platform for training and developing these architectures at scale, which enables us to modularly combine them to solve all sorts of different problems. So, okay, how does data and model come together to sort of inform the way that you're going to pick solutions to different problems in this space? So I tried to create this, I created this table to kind of guide your ability to navigate this. There's obviously a lot of room for interpretation and discussion in between these, but I wanted to just give some examples here, and I'll talk about some real world examples after. But just briefly looking at human interpretable features and the supervised computer vision models, you can think about them as high in interpretability, low in data requirements, and high in bias. And when I say bias here, I mean in terms of bias variance. So the reason that they're high and bias variance trade-off. So the reason that they're high in interpretability is because the features or their outputs are directly traceable to specific biological objects in the image. This makes them really great for enabling hypothesis generation and integration with sort of the systems or systems biology understanding of the disease that you're studying. The data requirements are low because the way this model is set up is such that the data set size is really the number of annotations that you generate rather than your number of whole slide images. Of course, we don't train human interpretable feature models in, say, 10 slides and think they're going to generalize well, but we can generate millions of annotations from a relatively small number of slides in order to train pretty good segmentation models. And then they're also biased, and that bias is driven by their ability to be low data requirements. So basically, because we pre-specify features, you pre-specify the biological objects that you think are important, that's going to yield a more biased estimator. It's just going to overall be fixed on the features that you predefine. The graph neural networks and the end-to-end models, those are both approaches for classifying whole slide images. And in terms of interpretability, they're about the same. They both would have moderate interpretability. I think our end-to-end models are actually more interpretable than the rest of the field, so that might be low for some other approaches. And the reason they're moderate is because the predictions are explainable, but they're only qualitatively really traceable back to the relevant regions and morphologic features that are driving those predictions. The model can highlight and tell you which features are important and where in the image those features are present, but it's not going to sort of give you a neat biological hypothesis in terms of lymphocytes without the intervention of a human pathologist looking at those areas. In terms of data requirements, the GNNs are more moderate, and they're also more moderate in terms of bias. And the reason for that is because we construct our GNNs on already featurized or biologically driven featurization. So in H&E, we might construct our GNNs on top of our cell and tissue heat maps, or we might apply that in multiplex immunofluorescence, where you already have the protein positivity that you're interested in. But that also comes at the cost of introducing a little bit of bias because you're pre-specifying the node features which are going to be important. We mitigate that a bit by enabling a data-driven spatial pattern and feature and refeaturization. But ultimately, that's going to be more moderate in terms of bias. And then if you look at the end-to-end models, typically the data requirements there are higher and the bias is low. And the reason for that is because you're discovering the features in a purely data-driven way without doing any kind of pre-specification. So this could be mitigated. The data requirements can be mitigated a bit by pre-training the models. And we also, but generally these are going to be really high power and flexible models leading to low bias. So the numbers that I have here are really just the minimum starting point. We when we're developing our products, we develop our products with much larger data sets than these. But I wanted to give folks sort of a minimum grounding so that they can think about when they're able to start approaching these problems. And then just to make that last chart a little more real, I thought it would be useful to just talk through some examples or just list out some examples, and I'll talk through them quickly. So here are 3 examples. The first example is using a multiplex immunofluorescence data set to identify a spatial phenotype or signature of response to an immunotherapy agent. That data set was 150 non-small cell lung cancer samples that were imaged using the Akoya 6-marker panel. We also had survival data and RNA-seq data that was used in downstream analysis. The approach that we took was a unsupervised graph neural network that learned that spatial representation, and then we used those for sort of classical survival analysis and integration with that molecular data set that I mentioned. You can check out the poster. We published this at SITC in 2022. The second example is identifying morphological biomarkers of drug response, again in non-small cell lung cancer, but this time using a monostain for CD8. We also had RNA-seq data, which we combined here to just analyze the data. The approach that we took was supervised computer vision models that exhaustively annotated and quantified all the cells and tissue areas of that image. We then computed spatially informed human interpretable features and used GNNs for immune phenotyping. So both the HIFs and the GNNs were used for immune phenotyping there, and that is a poster from AACR in 2020. These are all on our website. And then the last one is a molecular prediction tool to identify patients with HRD. Here we had a data set of 931 breast cancer slide images. This is actually data from the TCGA with corresponding genomic sequencing data. This is exome sequencing for HRD, or exome sequencing derived HRD status. We used the additive MIL approach there to predict the HRD status. And that was a poster also available on our website from San Antonio Breast Cancer Conference in 2021. Okay, so moving on to evaluation metrics. So when you're thinking about your evaluation metrics, you should always focus on how you're going to pick your metrics and your goals and making sure that they're relevant to the use case that you have in mind. So to give you an idea of like how you might think about that, you should think, okay, am I looking for translational insights or am I looking to try to make a diagnostic device? So if you're working with translational insights, maybe it's okay for your model to be pretty good at finding cancer, and there's some false positives and false positives or false negatives. And you're just really using this to generate hypotheses. You can use a lower fidelity model versus a diagnostic device. You really want that model to have much higher performance characteristics, because rather than sort of operating in a translational setting, might be impacting patient outcomes. Another thing to consider is, for example, in a molecular triage tool versus trial exclusion, maybe you want a different sensitivity characteristic. So if you're using a molecular triage tool, maybe you want really high sensitivity to be able to capture as many patients as possible. Versus trial exclusion, maybe you want to have a different balance between sensitivity and specificity to make sure that the right patients are getting getting the right treatments. If you're designing a test data set, You should make sure that the test data set and your evaluations are matching the context of use. So when you're evaluating, making sure that your cuts of the data address technical covariates. So do you have a test data set and an evaluation metric for each scanner that's relevant to your use case, for each lab and staining protocol that you might want to see that this model generalizes to? Are you also representing your biological covariates in those metrics? So for different disease severities or genomic statuses, do your models perform uniformly, or are they impacted by a different histology? And then finally, you should think about, especially in digital pathology, the types of consensus metrics that you want to use. You have to remember that pathologists are going to disagree. There's going to be, there's not really a ground truth. So you have to figure out how you're going to use consensus to define that ground truth, and what is the relevant consensus or the relevant metrics that you're going to see in the field. On top of that, you should think a bit about what types of qualitative and quantitative metrics you want to collect. So I've collected some examples here just to give sort of ideas for how to start thinking about it. But if you're doing pixel level classification, a qualitative metric that we use quite often is pathologist review of your model on top of whole slide images. We also use quantitative performance metrics as well, where we look at performance on annotations, and as well as looking at performance within exhaustively annotated frames. And we'll talk more about our frames pipeline in a later webinar. We also think about, for slide level classification, you have qualitative measures where you might have a pathologist interpret a heat map to see if the model is doing something quote unquote reasonable. And then you can also use sort of the very standard machine learning techniques for slide level performance metrics, like area under the receiver operating curve. And then for unsupervised learning, there really aren't a lot of good qualitative metrics. You can look at your images. For example, with the scanner transform, you can think about comparing the images to real images and looking to see if those look reasonable. In terms of quantitative metrics, you can, there's machine learning objective functions to see what the quality of your images are. If you're using a generative adversarial network or a GAN, you can see what the test set performance is there. And then there's also performance on unsupervised, on benchmarks downstream. And I think that's really the measure of the quality and impact of your unsupervised learning, is to see, okay, using this unsupervised learning technique, did it make a difference for my downstream models? Are they more generalizable? Were we able to get better performance using less data by pre-training? And really making sure that you're having those kind of like impact metrics concrete if you're going to go down this rabbit hole. Because it can be, yeah, you can unsupervised learn for a long time without having a lot of impact if you're not really focused on those benchmarks. Okay, so just to tee up and talk more about consensus, or tee up the webinar that's coming later and talk more about consensus, because I think this is really an important aspect of the field, because we're using pathologist annotations often. I wanted to just go through the pros and cons of different types of pathologist consensus for model evaluation. So the first type of pathologist consensus is to have separate ground truth and benchmark pathologist panels. So you have a test set, that's one group of pathologists, maybe there's 5 pathologists there, and then you have a benchmark panel, that's another set of 5 pathologists. This is really the most fair way to generate ground truth, to evaluate your model and then evaluate that ground truth against another set of pathologists. It's treating the model and the pathologist fairly. But of course, it's really expensive and it takes a lot of time and might not be practical for all approaches, right? You have to get a lot of pathologists to assemble both that ground truth consensus as well as that benchmark consensus. Another way to approach this is to leave in all the pathologists to generate the consensus and evaluate each of the pathologists against that consensus. The problem with, or the pro of that, is you're using the same ground truth to compare each pathologist and the model. The con of that is you're giving an unfair advantage to the pathologist in comparison to the model. Now, this might also be a pro depending on how you think about it, because it's ultimately a very conservative measure. You're going to underestimate the performance of your model. But overall, it's treating them differently. I would say that that's a con. Another type of consensus is to have a mixed model and pathologist leave one out, where you leave out a pathologist from the consensus, but include the model in the consensus. This is really easy to compute among the left-in scores. It's really easy to compute that consensus. It's straightforward to understand. The problem there, though, is that you're treating the model as a pathologist, and that might be biased in the other direction. You might not want to do that, and it might not be looked at favorably. Another way to think about this is to do pairwise consensus. And there you're really not defining a ground truth at all. You're just measuring the performance of each of the pathologists in pairs, as well as the model as another member of those pairs, along with all the pathologists. The pro of that is you're not computing a consensus at all. The con there is that it's a little harder to interpret the results, right? You don't have like sort of one measure that you can point to and say like, okay, this is the performance. You have a population of scores that you have to summarize in some kind of way. And what we think is the optimal or the best approach, given the costs and statistical properties, is to do a per pathologist, leave both out. So you have enough pathologists where you leave the pathologist out and you leave the model out as you're computing that consensus and computing the metrics for each pathologist in the model. And the way you're doing that is that enables you to do an apples to apples treatment of the model and the pathologist. You're sort of evaluating them in the same way. And the difficulty there is that you need to get enough pathologists, making it so that you can have difficulty in computing the leave out consensus, right? Because you're separating a pathologist out. Okay, so just to wrap up here, sort of combining data, model, and evaluation, all the different sections that we've talked about, I wanted to just give some considerations for how you can evaluate external vendors and evaluate solutions in this space. So in terms of data considerations, you should think about, is the vendor or is the approach, is it being generated in a single data source, or does the vendor have access to multiple data streams? Note that like really there's no perfect solution to this. Everyone's going to have data challenges. It's inherent to the space. But you should think about how they're approaching mitigating those data challenges. So some of the things that we do that I mentioned in this talk is around generative modeling or pre-training and leveraging our huge data sets that we have in-house and our data partners. But in general, partners should be thinking about this and should have answers for how they're approaching it. And I think really importantly, partners should, or external vendors should be clearly documenting the characteristics of the training data, including important biological and technical covariates of where their models and solutions were developed. This is important because it's giving you the ability to understand where will this model work, where won't it work, where won't it work, or where will you be using it at risk. In terms of model considerations, I think it's important that external vendors have a variety of models, enabling them to address a diverse set of use cases, and that they're thinking about the biological context and the way that this is going to integrate into sort of the environment that this model is being used, and how those so to make sure that those model architectures are really fitting that use case in a clear way. And I think one of the important aspects of that is what model interpretability tools are connected with those offerings. So, if you're using whole slide prediction tools, how are they interpreting those model explanations? Is it connected to human interpretable features? Is it just driven by attention? What are the ways that they're sort of unlocking those models and making it so that you can connect them into your biological hypotheses, or avoiding sort of spurious predictions? And then the last piece is around evaluation, which we wrapped the talk talking about. I think it's really important to make sure that the models are being evaluated according to the particular use case. So are they considering the important technical and biological covariates when they're constructing their train val test splits? Are they showing that performance clearly? Are they computing ground truth in a reasonable way? Are they using consensus in a way that makes sense given your use case? Have they thought through all the different considerations there? Okay, and with that, I will wrap up and take questions.
Great. Thanks, Amaro. We have a lot of questions to get through, so that's great. It seems like people are engaged. And hopefully not only will you be answering some of the questions now, but some of these may carry over into some of the other presentations that you'll be providing as part of this webinar series. So first question, I'm not going to be naming names. Most of these are anonymous. The first one is, can you give an example of quantitatively versus qualitatively traceable predictions?
Quantitatively versus qualitatively traceable predictions. So, okay, so I would say, I think that that's probably referring mostly to like weakly supervised learning. So in that case, you would have a model, let's say it's predicting HRD, and it's traceable or explainable in the way that the model is telling you what parts of the image are important for that downstream prediction. So you can use that using like an attention heat map, which is one of the standard methods in the field. People also, we also have a different solution to that using our ADAM-ML framework. And it's qualitatively traceable in the sense that you can have a human pathologist review those patches and say like, okay, this is what I think is important. You could combine that with other like gradient-based methods where you look at those patches and see what parts of the patches are most important to the model's prediction overall. That might be a way to introduce some quantitative measures. Another way that you can sort of make that more quantitative is to combine it with supervised computer vision models, where for, let's say you have some highly attended to patches or some patches that are assigned as important for different classes, you can characterize the human interpretable features from those patches and quantitatively compare patches between the positive class and the negative class. And that would be like an example where you're quantitatively tracing it all the way back to like, what are the biological objects that are different between these patches? So I hope that answers the question. There's like, yeah, there's gradient-based ways where you can go back into the model to make it quantitative, but I think ultimately the qualitative side is just working with a human pathologist.
Great. Next question is, how do you determine how much data is needed for training in each use case? How do you decide how much data to gather at a slide level, tiles, versus at a case level, different types of clinical cases?
Ooh, that's a good question. So I think in principle, at a high level, I think if you're operating in a setting where you're doing something translational and you don't want to necessarily be, or you're not worried about building a model that's going to work in many different use cases or like in many labs throughout the country, for example, you can have a smaller data set. You can assemble a smaller data set, and it's maybe not worth it to annotate as much data on those images. So I would think about like, where do you need this model to work? If you need this model to work in more places, you need to collect more data, and you need to collect data that's representative of that real world application. In terms of the number of annotations and thinking about like multiple slides from the same patient versus single slides from the same patient, I think it's really, it's an interesting question. I think, again, it's gonna, maybe this is an unsatisfying answer, but I think it has to do a lot with like just the use case. So, you know, if you're, let's say you're building a device to measure like major pathologic response, You want to make sure that you're capturing as much tissue as possible. Maybe that's like maybe that's multiple samples from the same patient, to really be able to quantify and have like high certainty in the model's application. Maybe the same would probably be true for like cancer diagnoses. Like, let's say if you're doing like prostate cancer biopsy, maybe you want to look at like 12 different images or something like that from the same patient. And then I think otherwise, one image is probably enough. There are factors like intertumoral heterogeneity, which impact the biology that's present in each slide. But those factors are also present when you're doing molecular profiling as well, like for DNA and RNA-seq. And we typically assume that one sample, while it might not always be representative, is representative enough at a population level to be able to do translational research and understand our patients at a population level. So
Great.
I think I'll end there, but I think it's a complete
we got a lot more to get through.
Okay, sorry. I'll try to go.
No worries. These are great. I'm sure that everyone on the call appreciates the great answers that you're providing, the in-depth answers. Another question here is regarding the impact of biological covariates.
Yeah.
How do they impact H&E analysis? Is it possible to reverse this learning that AI can predict biological covariates from H&E?
Great question. Yeah. So to answer the second part of that question first, yes. If you build an H&E model to predict sort of like nuisance variables, turns out they work really quite well. So we've done this in-house. I don't think we've published this, but like we predict things like, I don't know, patient BMI or like tissue source site from TCGA images. So you can show that like not like those models are able to predict whether this tissue was sampled in Cleveland or whether it was sampled in Pittsburgh. So that works really well. And that's surprising. Humans can't really do that. And the question, the first part of the question was, I guess like there's two parts. One, you can protect yourself against having your model be influenced by those factors. That's where our generative learning and distributionally robust optimization methods that we use in-house come into play. And you can also tell if you construct experiments the right way to see if your model is being confounded in that way. So for example, one experiment that we've done in the past is we tested an algorithm that we were developing in-house on just background tiles to see whether it could perform a molecular prediction task. So we fed it just like literally white patches from all the different slides. And it turned out it actually worked quite well. And what that told us was that actually there's high frequency signal in the images that was being generated by the scanners that was enabling the model to cheat and do this kind of molecular prediction without learning any real biological signal. And we were able to do that experiment, identify that kind of like spurious signal, and then introduce training tricks to mitigate it.
Right. Yeah. Another question here. Sorry. How do you deal with inter-observer variation and annotation data in the training data set?
Yeah, good question. So couple of things. One is that we are always on our platform evaluating the quality of our pathologists. So we have in-house pathologists that review our network pathologists' work and QC those annotations to make sure that our pathologists in general are creating good annotations. In terms of how to address those inter-observer variabilities during training, we actually think it's kind of okay to have noisy labels. The one thing that we definitely do is try to source annotations from many pathologists rather than having all of our annotations generated by a single pathologist. And we hope that creates a robust data set that's not going to be biased in one particular way or another. So as long as it's not biased sort of directionally in terms of like classes, we hope that the gradient descent will just kind of work out by having random batches. And we do address this, however, in model evaluation by collecting consensus in those exhaustive frames and using the consensus methods that we had talked about to evaluate our pathologists and our model sort of jointly.
Great. We have time for a few more here. One is regarding the consensus and the established standards of practice consultation. How does AI developed by one company compare concordance with other companies' AI algorithms?
Oh, it's an interesting question. Like, I guess the question's sort of getting at, like, imagine a world where we have 3 methods that are all doing the same pathology task. How would we compute consensus among those 3 AI devices? It's a really good question. Nobody has done that because we don't live in that world yet. I imagine we would want to treat them the same way we treat human pathologists, though. So I think a lot of the same statistics would apply. I don't think we would do anything like, maybe, I don't know. I don't think we would do anything fancy where we're like tying the models together and ensembling the approaches or something like that. That would require the AI companies to really work together to make that solution. I also don't think it would be like easy to validate necessarily. So yeah, I think just the way you treat humans, doing the same thing.
All right. Are any of the model architectures available to the public or through a PathAI product to support development?
So in terms of code availability, our additive MIL model is publicly posted on GitHub. You can go check that out. And it's also, we published that method. In terms of none of our other methods are published in terms of code and to be able to use them yourself. But yes, we partner definitely with pharma companies all the time to develop products, and would love to make our architectures available to you to develop those different products. We can figure out different ways to do that. For example, if you wanted to use our artifact model to plug into your product, we can figure out a way to help you leverage that sort of like box of that modular framework. Or if you want to train your model in a scanner generalizable way, we can figure out how to partner and enable you to do that.
Yeah. Here's another question. Breaking down the process into 3 parts, how much of pre-analytical work is needed to implement specific AI in a specific lab to account for lab-specific staining characteristics?
Yeah, it's a good question. We hope, our aim is to minimize that amount of pre-analytical work and create models that will work as long as people are using the same sort of validated protocols. So we do this by using some of our robust optimization methods so that our models see lots of different types of staining results and other sort of like pre-processing procedures. But ultimately, I think it's going to be, yeah, I mean, we're going to try to validate and make sure that we can solve that from a method side to make adoption of AI not require people to sort of change or level up the uniformity of those like pre-processing steps.
I know we're coming to a close here. Just a couple of things. One was more of a comment about learning more about cheating AI, as you referred to it. And someone's very interested in learning more about that. I think that was a sort of a trigger term for me as well. Obviously, some neat capabilities in there and things that we can learn from. So definitely something that maybe we can incorporate in the future. The other is to add on to the question earlier, which is it possible to be a CAP requirement, like laboratory validation and verification? Would love to continue this one-on-one. I'm not sure who this person is, but having a hard time deciphering that one. Not sure if it meant anything to you or if the person wants to clarify real quick.
Just while you're while they're maybe chatting, I would say, like, if you're interested, so I think this like cheating issue around AI models is really worth digging into. And if you're wanting to read a good review, I cited a review, or it's not really a review of cheating per se, but it's about causality in machine learning. This Nature Communications paper, Causality Matters in Medical Imaging, is a really good primer on how to go wrong, or like how these things can really go off the rails. If you want to check out that paper, I think it's well worth a read.
Great. Last question before we end. You mentioned not using a trichrome type stain, but inferencing from an H&E slide for fibrosis. Why?
Oh, I don't think that there's necessarily a good reason to do that. You could definitely use trichrome staining. I was just mentioning it as an example of something that we could do where we're taking our generative model and adding it into our products pipeline. This is not something we do now. I think, IHC is and trichrome staining is like part of clinical practice. There's no reason to disrupt that. You get a lot of great data from that as long as those images are easy to generate. In a world where like artificial staining worked really well and maybe it was really hard to get extra tissue, like let's say you just had H&E slide images and you wanted the trichrome, maybe that would be a use case, but I think in general it's sort of limited in utility.
Great. Well, we're at the top of the hour here. This was extremely productive. I'm glad we got to get through all of those questions. And Amaro, can you go to the first slide real quick in your deck? Just wanted to thank everyone today, especially Amaro for taking the time out of his very busy schedule to be able to present all this to us. Looking forward to the next webinar on February 15th, which is really going to dive into the graph neural networks piece. And so I hope everyone here can join us and look forward to the next discussion.
Cool. Thanks, everyone. Have a good rest of your day. Bye.
Thank you. Bye.